All right, let's get right into it. We usually think of our DNA as this static blueprint, right? A fixed set of instructions for building a person. But that's really only half the story. The real magic isn't just in the code itself, but in the incredibly complex operating system that reads it. It's a system that tells our genes when to turn on, when to shut off, and exactly how loud to play their tune. Today, we're decoding that operating system.
So get this, just 1.5%, that's it. Only 1.5% of our entire genome actually contains the code to build proteins. The fundamental building blocks of our bodies.
Which, of course, raises this massive question. If only a tiny fraction is the blueprint, what on earth is the other 98.5% doing? For decades, scientists kind of dismissed it, they called it junk DNA, but we now know it is anything but junk. It's the control panel, it's the software, it's the conductor leading our entire genetic orchestra.
So in this explainer, we're going to pull back the curtain on that control panel. We're going to look at the local switches, hardwired right into our DNA, the remote control molecules that manage genes from across the cell, the secret regulatory life of RNA, and finally, what all of this means for the future of medicine.
All right, let's get started. We're moving beyond that simple idea of a genetic blueprint and diving straight into the dynamic world of the control panel, the system that actually executes the code. We're talking about gene regulation.
First up, let's look at the most direct form of control there is, the switches that are physically part of the DNA strand itself. These are called cis-acting elements.
Okay, so what does cis-acting even mean? Well, cis basically means on this side. So a cis-acting element is a stretch of DNA that can only influence the genes on the exact same piece of DNA it's physically attached to. It can't jump over to the other chromosome, the one you got from your other parent, and start giving orders. Its control is strictly, and I mean strictly, local.
The best way to think about it is like this. A cis-acting element is like a dimmer switch that's physically hardwired to one specific lamp in your house. It can only control that one lamp, period. Now, we'll get to the remote controls in a minute, but this idea of local hardwired control is absolutely crucial for understanding how our bodies fine-tune everything with such incredible precision.
And here is a fantastic, real-world example of this in action. Before we're born, our blood uses a special fetal hemoglobin to grab oxygen from our mother. Right around birth, though, a switch is supposed to flip, turning off the fetal gene and turning on the adult version. That switch is a cis-acting element. Well, in a condition called hereditary persistence of fetal hemoglobin, or HPFH, a mutation breaks that little switch. And the result? The body just never gets the memo to turn off the fetal gene, so adults keep on producing it. It's a perfect illustration of how one tiny change in a non-coding switch can fundamentally change a critical biological process.
So, if cis elements are the local, hardwired light switches, what about the remote controls? Well, that brings us to transacting elements. These are the regulators that can manage genes from basically anywhere in the cell. A transacting regulator is a molecule, usually a protein or maybe a piece of RNA, that's made from a gene somewhere else in the genome. Then, it floats through the cell to find its target. Because it's a free-floating molecule, it isn't tied down to one chromosome. It can regulate the gene on the copy you got from your mom and the copy you got from your dad. It's like a broadcast signal, not a hardwired connection.
And here is a beautiful example of this system at work, how your body manages iron. It's this elegant feedback loop. When your iron levels get low, a transacting protein called IREBP kicks into gear. It does two things at once. First, it physically blocks the production of ferritin. That's the protein that stores iron because, why store it when you need it? At the same time, it stabilizes the instructions for making the transferrin receptor, the protein that absorbs iron from your food. So you store less and you absorb more. And when your iron levels are high again, the protein is inactivated and the whole system just reverses. It's a brilliant remote-controlled system that makes sure you always have just the right amount of iron.
For the longest time, we thought of RNA as just this humble messenger boy, just carrying instructions from the DNA to the protein-making factories. But it turns out, RNA is a major player in the control room itself. It has a secret regulatory life that we are just now beginning to understand.
This is honestly astounding. While only 1.5% of our DNA codes for proteins, something like 70% of it gets transcribed into RNA. Just let that sink in. The vast majority of RNA made in our cells isn't carrying messages to build things. Its job is something else entirely, regulation. Welcome to the vast world of non-coding RNA.
And to really get a sense of their power, we have to look at one of the most mind-bending phenomena in all of genetics, genomic imprinting. We've got two very different neurological disorders here, Prader-Willi and Angelman syndromes. What's absolutely wild is that they can be caused by the exact same piece of deleted DNA on chromosome 15. The only thing that determines which disease a child gets is whether they inherited that deletion from their mother or from their father.
Okay, so here's how it works. Let's start with the chromosome you inherit from your dad. On that copy, a special gene called SNRPN is switched on. Now this gene doesn't make a protein. It makes what's called a long non-coding RNA, or lncRNA. A key job of this long RNA is to basically stretch out across the DNA and physically block another gene nearby, called UBE3A. So, on your paternal chromosome, UBE3A is always silenced.
But on the chromosome you get from your mom, it's the complete opposite. The lncRNA gene is off. That means there's nothing blocking the UBE3A gene, so it's active and working. Now, this is so important because in our brains, only the maternal copy of UBE3A is actually used. So you can see what happens, right? If you inherit that deletion from your dad, it's not a huge deal for UBE3A. But if you inherit it from your mom, you have no working copy of this critical brain gene, and that leads to Angelman syndrome. The entire thing is controlled by a non-coding RNA.
And if you thought that was complex, there's another whole layer of these things, tiny little RNAs called microRNAs, or mRNAs. And these guys are everywhere. We have over 1,000 different kinds, and collectively they are thought to regulate about 1/3 of all our protein-coding genes. They act like precision tuners, dialing down the output of specific genes.
Check out this fantastic comparison to see the difference between tuning a gene and just turning it off completely. On the left, you've got the Texel sheep, famous for being extramuscular. This is caused by a single, tiny mutation that accidentally created a docking site for a microRNA on the myostatin gene. That's a gene that normally puts the brakes on muscle growth. This mRNA tunes down myostatin, and the result is more muscle. Now, on the right, you have the Belgian blue cattle. It has a mutation that completely breaks the myostatin gene. The brake isn't just tuned down, it's gone, and you get this extreme double-muscled look. This just perfectly shows how a subtle RNA regulation can create a useful trait, while a brute force mutation has a much more dramatic effect.
So after that journey through all these different layers of control, where does this leave us? Well, we are standing right at the edge of a new frontier, where understanding the genome's control panel is becoming every bit as important as just reading the code itself.
We've really peeled back the layers of this incredible operating system, haven't we? We've seen the local, hardwired switches of the cis-acting elements, the mobile, remote control proteins, and this vast, influential network of regulatory RNAs that act as spies and fine-tuners, directing the whole operation. It's a system of truly breathtaking complexity.
And this really is the future. As this quote from our source material puts it, understanding how gene expression is restricted in time and space, how a gene is turned on in a liver cell but not a brain cell, or at age 5 but not at age 50, that is the next great challenge, the next frontier in genetics. So here's the bottom line. We've moved way beyond the blueprint. We're now exploring this vast, dynamic control panel that operates our DNA. It turns out many diseases aren't caused by a broken gene, but by a broken switch. And as we learn to read and maybe even rewrite the logic of this system, the question becomes, what new therapies, what new cures are waiting to be found, not in the 1.5% that makes proteins, but in that other 98.5% that's been running the show all along?